Okay, thank you so much, guys. I appreciate it. So today we're doing lecture 8. We're well into material for midterm 2. We did 2 more lectures and then we're done again in another exam. Okay, so muscle structure today. I think one of the biggest problems with the modules was just knowing the structure and the function of those proteins that are involved in muscle contraction, and also how the energy is used up for the muscle to contract. Such as when the myosin thick filaments attach, there's energy that's needed, and then when they pull back and retract the other actin filaments inwards, energy is used, and to break the bond between the myosin and actin, energy is used too. Okay, so we'll talk about that and talk about how the muscle is compartmentalized into bundles using connective tissue. All right. Okay. Good. So, here's the functions of skeletal muscle. It's very intuitive, really. So, movement, why? Because the skeletal muscle is attached to the skeleton. So, the skeleton, when they act as the lever, and then the muscles, when they contract, they move the levers, and then there's movement. Because muscles contract with the force that can propel you, and so forth. So, skeletal muscle. So if I ask about the functions of muscle, I want you to kind of tell me what the muscle type of muscle is involved and how it contributes to that function. Okay? So here you have to explain the attachment to the skeleton and how the skeleton acts as the levers and the contraction uses movement, then causes movement. And then maintenance of posture. Right now, as I'm standing up, my quadriceps are contracted and my ab muscles, my glutes, all that to maintain my body straight. Okay, so posture and skeletal muscle, because it's attached to the skeleton, right? And what else? Respiration. How are muscles, skeletal muscles, involved in respiration? These would be muscles attached to which bones? For respiration? Yeah, ribs, intercostal muscles, right? The intercostal muscles, when they contract internal and external intercostals, they will expand the chest cavity, and then you suck in air, and then contract the volume of the chest cavity, then you push air out, you expand. So that's how it's involved in respiration. Okay, and the diaphragm itself is a skeletal muscle too, and it contracts like a plunger, just like a syringe, sucks in air and pushes air out. Okay, so if I was to get you to explain that function, you should be able to explain it, no problem, right? Respiration and then the skeletal muscles that are attached to the ribs. Okay, you don't even have to say intercostal muscles. You just say muscles attached to the ribs. That's good enough. And then they expand and contract the chest cavity for inspiration and expiration. Right? Production of heat. Okay. One of the things is that there's no 100% efficient machine. So when the muscles use the energy in ATP, they do not use all of the energy. So whatever is excess heat, whatever is given off, that's what is used to heat the body. And then the blood distributes it around, So muscles, ATP energy, contributes to body heat. Body heat temperature, okay? So in the exam, don't just say body heat and then stop there. Tell me how that happens. All right, don't just say posture. Okay, tell me how that happens. Or movement and things like respiration and then stop it. You gotta tell me further how that muscles are contributing, which muscle too, skeletal and all that. For the most part, because skeletal muscle makes up about 40% of your body, it's gonna have most of the functions, right? So communication. Right now, what I'm doing, I'm talking, right? How else are muscles involved in communication? Skeletal muscles, that is. What's that? Facial expressions, yeah. When I frown, you know that I'm not really happy right now. When I'm smiling, then you know that I'm all good. Right now, what are you doing? You're writing, typing. All those is communication with skeletal muscles. Okay? When you look at communication, you think, what the hell does skeletal muscle have to do with that? But there's a lot, right? There's a lot of stuff that you can talk about, just communication and how skeletal muscle is involved. All right, so and I want you to do that in the exams. And you guys are good at it, so keep it up. I'm almost done with your exams. So, constriction of organs, that would be smooth muscle, right? What are some of the organs in your body that have smooth muscle in them? Yeah, the esophagus, the intestines, all that digestive. What else? Another important one. Well, if people say the blood, I don't know why the blood. Is it because when you're not gonna go, your blood is all full? I don't know. But the blood vessels. Blood vessels have smooth muscle in them, and they distribute blood, nutrient, oxygen throughout. So that's a very important smooth muscle right there. Okay. So if you ever talk about the function of smooth muscle in those internal organs and all those other organs, be sure to mention blood vessels because that's an important one. Okay, passing oxygen and nutrients throughout. So constriction of organs and vessels. So blood vessels is pretty big on this one. So be able to blood vessels. Okay, smooth muscle in blood vessels. And then there's only cardiac muscle in the heart. It's the only place where you find cardiac muscle. And cardiac muscle is very special. It pretty much takes the best of the skeletal and the best of smooth muscle and puts it together. And you can imagine why that is, right? Why would cardiac muscle do that? It's the best of skeletal and the best of smooth. Why? Why only in cardiac muscle? Right now, when you're sitting down, your skeletal muscles are taking a big rest. What about your heart? Your heart is still beating inside of you right now. Even when you go to sleep, your heart is still beating. So your heart never rests. So it needs to be very specialized. So you will find specialized structures in there like intercalated discs and so forth. And it's striated just like skeletal muscle. And it acts almost like smooth muscle as well. Okay, so it kind of takes the best of both. So cardiac muscle is only found in the heart. And of course, it produces the force that distributes the blood around the body. Very important, right? So be able to explain these functions in the exam situation, right? Don't just say heartbeat and then stop. And then I'll just give you half a point and stop. Okay? So I won't go further than that. All right. So because muscles have certain attributes, which we're going to call characteristics of a functional nature that allow the muscle to do something. Okay? So these are functional characteristics. They can contract, they can extend, they can recoil, and so forth. Okay? So the contractility is a functional characteristic because it can contract and produce a force when it does that. Right? So it's a functional characteristic, right? Because it is an end result. It is an action that comes with that characteristic. All right. So the contractility is the ability to shorten forcibly and produce a force that can propel me to walk forward like I'm doing and do stuff like that. And then there's no point in a muscle being able to generate all this strength when you cannot excite it, right? It's like, when you're trying to talk to your friend and your friend is busy trying to ignore you. Busy, there's not going to be any connection. What I'm saying? It's not going to be a good unit. So muscle, because it has the ability to be excited, you can send a nerve impulse to it and then it will respond by contracting forcibly and producing a force. Okay? So being able to be excited is an important function of muscle. All right? Being able to respond to stimuli. And then later on we'll be talking about how some muscles can self-excite like the heart and some smooth muscle. But skeletal muscle for the most part is voluntary control. You have to think about actually contracting the muscle for it to contract. But there are instances when it's involuntary control. And it can extend. Okay? The muscle can be stretched in length so that when it recoils back, it will produce the force. So the ability to be stretched or extend is also important because then it's elastic. It can recoil back. You stretch it. If muscle, is able to extend and it extends and it extends, and then it stays there, you get problems. It has to come back to original shape. That's when it actually produces the force, when it's getting smaller. Okay? So elasticity. So those four things if I talk about the functional characteristics of muscle, sometimes in the exam I'll ask about it. So you talk about extensibility, contractility, excitability, and elasticity, right? So, and then you can talk a little bit further about it as to why that function is, that characteristic is an important function. All right, so now types of muscle tissue. We kind of covered this when we were talking about tissues. Remember last time? And I said, forget about muscles, forget about the nervous system, because we are going to have separate lectures only talking about that. So here's one for the muscles. Okay? So we have the smooth, the skeletal. Smooth is called smooth because it is, you know, it doesn't have striations like the skeletal, I mean like the skeletal muscle. All right? And then of course cardiac is the one that's only found in the heart. It is also striated like skeletal muscle, but it has some characteristics that are also similar to smooth muscle. And I'll show you a table with comparing all these three muscles, because I'm going to need you to know the differences between these three structurally, functionally, and location-wise, and so forth, which is what it says right here. They're going to be different structurally, like for the example, skeletal muscle, very long cells. So long, we call them muscle fibers instead of just calling them muscle cells. Because they're that long, they have a bunch of nuclei inside. One nuclei is not going to be able to serve that very long cell. You know what I'm saying? Close it back up. And so that's important. Location. We just talked about skeletal muscle. It's located where? On the skeleton, right? And the heart is only found, cardiac muscle only found in the heart. And then of course smooth muscle in the what? Smooth muscle is found where? Digestive organs and blood vessels, right? Okay, and then means of activation. We talked a little bit about it, saying that skeletal muscle is for the most part voluntarily controlled. A few instances when it's involuntarily controlled. The other two, cardiac and smooth, are involuntarily controlled. Also, smooth muscle and cardiac muscle will respond to hormones, which skeletal muscle don't. Because hormones can regulate heart rate, things like that. Hormones can regulate your smooth muscle, okay? So they respond to hormones as well. And they have the ability to self-excite, like the heart actually has special cells called sinoatrial node, or sometimes called pacemaker, which can self-excite without any external stimulation. It can self-excite. So, so means of activation and so forth. So, and we'll talk about these differences in the table that I was talking about. All right. So each muscle is a discrete organ. We know the muscular system, like the biceps, is an organ of the muscular systems. All right. And then what do you need in the muscle? Of course, you need the muscle tissue, right? And you need the nervous tissue to do what? What does the nervous tissue do in the muscle? It's excited. That's what brings the stimulus, right? And then the important thing also is the connective tissue. You need the connective tissue in there, because what muscle is like a bundle of fibers, all divided up by. That is that you want guys and all hands or whatever. So, and it will be like a bundle, like let's say these are muscle cells, and then a bundle like this would be like a fascicle, and then it will have a perimysium surrounding it. And then each muscle cell or each muscle fiber, because the plasma membrane is very weak, right, for something that is contracting and being pulled apart, it is also covered by connective tissue called the epimysium, right? And then the whole muscle bundle, like the whole bicep, is covered by the epimysium. Right? And the epimysium is continuous with what? The one on the outside. With the tendons here, because then that's how the muscle is attached to both. Yeah, the outside. So this connective tissue is important. In fact, we'll talk about it, in terms of how it's compartmentalized, some of the other functions of connective tissue. So this is the skeletal muscle showing the bands, because the skeletal muscle again has the fibers inside of it, like the actin, which are the thin filament, and the myosin, and when they are arranged, they have dark and light bands because of their overlap and so forth. Okay, so that's what creates the striation in the skeletal muscle. Also in the cardiac muscle again there, which you can see the differences now. Smooth muscle is like that, it's smooth. It's really got no striations whatsoever, and the muscles are short, and they have one nucleus, also cardiac muscles are also short in the whole nucleus, and we'll talk about the significance of that in a little bit. So, types of muscle tissue. So skeletal muscles, of course, so called because they are attached to the skeleton. Okay. Don't just say, sometimes people forget to put the muscle on there and they just say skeletal tissue. If you say skeletal tissue, we're talking about bones. See what I'm saying? So don't forget that. So, so if you're gonna leave anything out, it's the muscle part. Leave the skeletal part, at least. But because if you say skeletal, then you're totally in another system and you're talking bones this time. So skeletal muscle, they hold that because they're attached to the skeleton. And the functions we just talked about is the movement, the posture, and so forth, right? And of course, muscles generate heat because when they break down energy from ATP, they don't use up all the energy. The rest of the energy is given off as heat that is used to maintain body temperature. All right. So smooth muscles, very important. Like I said, the blood vessels. The blood vessels have smooth muscle in them, and so it helps to maintain the blood pressure. All right. And then also the digestive system. All right. So this would be the vascular system. All right. And this squeezing and propelling stuff is the digestive system for the most part. Like your esophagus, stomach, the intestines, the colon. Okay, so moving waste and food along. Waste by the time it gets to the colon in terms of feces and stuff. And then, of course, cardiac muscle, only found in the heart. It's got very specialized connections between the cells called intercalated discs, and we'll talk about them when we discuss the differences. All right, and this supplies the force that distributes blood around throughout the body. The heart does. It's a pump. That pumps blood. So this is the table I was talking about. I'm going to need you to really know these differences because sometimes I'll talk about you discussing the 5 differences between skeletal, smooth, and cardiac muscle. So the location is obvious, right? The skeletal muscle attached to the skeleton, right? Cardiac muscle only located in the heart, right? And then of course smooth muscle in the soft internal viscera, like blood vessels and the digestive system. So that's easy. Appearance, we talked about it. The skeletal muscle cells are so long that we call them fibers because of their length. And because of their length, they have a bunch of nuclei, so they're multinucleated. And the nucleus is located on the periphery, okay? Right on the outside of the cell. So this will be the cell right here, and then nuclei will be located here, the cell on the outside. Why do you think that is for skeletal muscle? What's that? Yeah, but what is inside of the cell? Inside of the cell, you've got these fibers, the actin and the myosin. You know how they do this? Slide past each other and contract. So imagine if the nucleus slept right in the middle of that network. It wouldn't work. You know what I'm saying? So that the nuclei are out of the way, they are on the periphery, on the outside of the cell. So that the contractile mechanisms of those proteins, sliding past each other, it doesn't have like a big bump of a nucleus in the middle, and then it won't work. See what I'm saying? All right. But the fibers in this are not striated, they are not arranged in any fashion. So that's why it's smooth muscle. So these are spindle shaped, okay? They look like a spindle, like this. And then with a centrally located nucleus. You see what I'm saying? And inside of the skeletal muscle, you have the myosin and the actin filaments here arranged. That's why you see these bands of dark and light along the skeletal muscle, giving the striations. All right. The cardiac muscle is short, it's branched, and it's got one central located nucleus as well. Why? Because the cardiac muscle contracts in like different shapes and forms. Like even the heart shape, the heart is kind of like that, right? So when it contracts, it's almost literally like it squeezes the blood out of the ventricles. So in order to be able to contract that way, you need the fibers branching in different directions so that it can contract like that in a fashion. Plus, the muscles, cells here, the cardiac muscle cells are shorter, so, and they are connected by intercalated discs, which have gap junctions in them. Okay? You know what a gap junction is from the last time? What is a gap junction? Yeah, allows especially the impulse to pass through all the cells, because the cardiac muscles need to contract like one muscle cell, and there's many of them. So they are shorter and they are connected by these gap junctions, which is pretty much a protein channel. That a channel made of protein on which ions can enter when the membrane potential is changing. Okay? So single located nucleus, central located single, but multiple, and on the periphery on the other side. Right? So striations, of course, because of the arrangement of the fibers, yes. No for this, because the fibers are all over the place. And also, these are connected by gap junctions as well, so that they contract as a one functional unit. Okay? And these ones, they also contract as one functional unit with intercalated discs, which are specialized gap junctions. Okay? These are just simple gap junctions, but for the muscle, for the cardiac muscle, it's special gap junctions, only found in the heart. Okay? So, like we said, voluntary control for the muscles and involuntary for smooth and cardiac. In fact, some smooth and cardiac muscle can self-excite without any external stimuli. Remember, I said there is some instances when skeletal muscles are involuntarily controlled, like reflexes right now. Like the upright reflex for me right now to stand upright, it has to contract certain muscles on my body, relax certain muscles, so that I am standing upright. And if I have to think through that, I'm trying to teach you and talk to you, I'll be kind of confused. In fact, I won't be good enough to do that. So some of those functions, you know, are automatically controlled and it's reflex. But for the most part, skeletal muscles are voluntarily controlled. Like right now, if I was walking barefoot and I step on a tap or a broken glass, what am I going to do? Immediately, I'm going to withdraw that leg, and then my weight is going to have to pass to the other side. Imagine if I was thinking about that myself. I wouldn't be good enough. I'll be too slow for starters, you know, and I won't know which leg to apply to take over my body pressure. So all that happens automatically to limit tissue damage. So those are the few instances when skeletal muscle are involuntarily controlled for spinal reflexes. Makes sense, right? Because I wouldn't be able to handle that. No, I may be bright, but not to that extent. Okay. Yeah, that's how it is. So that's that. Okay, but for the most part, it's voluntarily controlled. And then, of course, capable of spontaneous contraction. No, yes, yes. Right? And then, of course, the function, you know about body movement, we know about the pump, the heart muscle, and we know about moving foot. So in the exam, you should be able to remember at least 5 of these things and be able to tell me the differences between skeletal muscle, cardiac muscle, and smooth muscle, right? Pick the most glaring ones and you should be able to come up with 5. And then maybe come up with 2 more for bonus points, right? Again, our little magnification gets higher for the striations. So the striations form the A band, the I band, and I'll show it to you in a little bit. So pretty much what happens here is that you got the Z line here. In fact, the functional contractile unit of muscle is the sarcomere, and I'll show you. So on the sarcomere, you've got these fibers there, okay? And then you've got the thick filaments. These are the actin thin filaments, and then the thick filaments are like that in there, and they're wedged here at the M line by desmids, proteins called desmids. So what happens is that the part where you just have the actin, like from here to there, that's what is the I band, right? And the part where you have the myosin, that's what is the A band, okay? And you can see how the actin, which are thin filaments, will be light, and I'll show you next pictures later, so don't worry, okay? And then these thick filaments will be the darker bands because they are thicker. And you can imagine the part where the light and the dark bands, interlaced, or where they you know, come together, will be even darker, right? Because you've got the light and the dark together. And this part here, which we're going to call the edge zone, okay, just where there's just the thick filaments by themselves from here to there, you can see it's going to be slightly darker than the part where there's light. So those are what creates those bands, okay? And so when the muscle contracts, the sarcomere will shorten because it will come, the Z lines will come together, okay? And then you start to see the ident disappearing, the edge zone disappearing. Okay? Those are some of the things we have problems within the module in terms of showing which of these bands gets shorter when the muscle is contracting. And we will really go into that detail. So I wanted to kind of preempt it by showing it to you now so that you can see what is accounting for these bands going back, you know, dark and light and so forth. It's because of that arrangement of the actin and their overlap. Okay, see where they overlap here, where the interlaced will be the darkest bits because you've got light and the dark bits together, right? So, yeah, like we said, you know, skeletal muscle cells are so long that they are called fibers, skeletal muscle fibers. Okay, just because of their length. And that also is the reason why they are multinucleated. Right? Because just one nucleus won't be able to serve the whole cell. Okay, so you have a lot of nuclei. All right? And we said they're located on the periphery. Why? Because those interlaced actin, thin filaments, myosin, thick filaments, if we had a nucleus in the middle of all that network, it wouldn't work properly in terms of them sliding past each other like this, nice and smooth. You know, it would be going like it won't work. So the nuclei to be out of the way, they are on the outside. They are on the periphery. Okay. So in the exam, if I was to ask you why is it that the nuclei in the skeletal muscle fibers are located on the periphery or on the outside, you would say? What would you say? So, yeah, so they will describe the sliding mechanism of the actin and myosin, right? Which we're going to call the sliding filament model, right? And we'll talk about that towards the end when we start to talk about which of these bands is getting shorter and so forth. Right. So the fibers, because of that, how the actin and the myosin are arranged, appear striated. Okay, so that's so skeletal muscle is striated because of the actin. These are the thick filaments, and these are the thin filaments. You know, the actin starts with an A, so it's the shorter, the thinner one, and it starts with an M. Myosin is the thicker one. All right. So. And the actin is a little bit more involving than just having actin protein. Okay, it's gonna really be involving. There's a few more proteins that are involved with it, and we'll talk about it. Okay, you know, so because they are long fibers, right? A single muscle cell, a single muscle fiber can actually span the length of the muscle. Okay, for the most part, they overlap, so that it's nice and strong. Right, but it can happen that the length of the muscle is spanned by the muscle fiber, muscle cell. But you know, skeletal muscle, the good thing about it, contracts rapidly and with a great force, but it tires quickly. Okay, it tires quickly. Cardiac muscle has no time to be tired. The way it gets tired, you're pretty much dead right now, right? When your heart stops, you stop too. So cardiac muscle, the only thing it does is there's a longer refractory period, a period that it won't respond to stimuli. So it kind of rests that way, but it's contracting all the time. But skeletal muscle, because it has the ability to rest and you can rest it out when you're tired, like if you're working out at the gym, when you've done your last time, you put the weight off. Okay, you don't keep trying to, otherwise, you know. So contract rapidly, tire easily, and mostly voluntarily controlled. Few instances like we talked about, when like reflexes, when it is controlled automatically, okay? But for the most part, it's voluntarily controlled. Right, so skeletal muscle structure. Remember all those connective tissues I was talking about, the epimysium on the outside, the perimysium covering the fascicles, which are like sub-bundles inside, and then the endomysium covering the actual muscle cell. Remember, because the plasma membrane is got that phospholipid bilayer, and it's not strong enough to sustain the kind of contraction and pulling apart the muscles are, are put through. So you have the endomysium, which is a tough connective tissue. All right, all those connective tissues are called fascia. That's what they're called. Okay, all those connective tissues are called fascia. All right, and those are three of them. So the epimysium is on the outside, is the overcoat in this dense collagenous tissue that is continuous with the tendons. The tendons attach muscle to bone. Right? So covers the entire muscle like the entire biceps, okay? Which has got two heads, two subbundles. Right? And then the perimysium is got it's like bundles of fibers like that. Fascicles with cells inside, all covered by the epimysium on the outside. And then the green will be the perimysium covering the bundles of muscle fibers, and then the red will be the endomysium covering the actual muscle cell. Okay, because we said the plasma membrane is just too flimsy. The fluid mosaic model with those lipid bilayer cannot sustain that, force. It cannot withstand the amount of force the muscles go through when they contract. So, gotta know these connective tissue membranes and which part they pretty much compartmentalize. These muscle fibers, bundles of muscle fibers put together. Okay, so one of the big things about connective tissue that we saw with the bones as well is that what does it do? It, like on the bones, remember on the bones we said the bone has got a connective tissue membrane, and what did you say runs on it? On the connective tissue on the outside of the bone? The blood vessels. Same thing with the connective tissue in the muscles. The connective tissue in the muscles also provides a pathway for blood vessels. Same thing, okay? So the thing about the physiology is that when you learn it in one instance, the same tissue will be doing the same thing in another instance if you apply it to a different situation. Same thing. So it's not really like learning. So that's why, I was telling the guys in the morning, the information we learned in the first exam, midterm 1, all those things we talked about, all those tissues and all those functions of cells. Right now we're talking about muscle cells. And the organelles in muscle cells are specialized, actin and myosin. Actin and myosin are nothing more than just organelles, and the organelles differ depending on the function of the cell. Like the muscles are specialized to contract, so it's got these protein fibers inside of them called actin and myosin, which are simply organelles. There's nothing special about them except that they are specialized for contraction. Okay? So the functions kind of have to continue, and you have to remember all the basic information we learned at the beginning as the foundation, on which we just build on top of as we go. So definitely pathway. You see how on this perimysium we got all these blood vessels running on it, just like we found in the bone. So that's another important function of connective tissue. All right. So so right now I'll just bring your attention to the actin and the myosin. Okay. The big thick ones, there's nothing much going on them except these heads called the myosin heads that form cross bridges when they are attached to the actin. Okay. And this sort of golf club looking like bundles. And that's it, and then it's got a hinge and a head. Okay. But the thin protein, the actin, has got a lot going on it. Okay? So we really have to learn the proteins on it. You see how the actin molecule looks like if you take a string of pearls, like a necklace of pearls, or Mardi Gras beads, So, and you twist them around like this. That's the exact structure of actin. Okay, and the beads inside of it will be what we call G-actin, which is globular actin. That's all that is. Okay, so this is G-actin globular. What is it doing? I don't know. For some reason, my board is now not working. Is it flash? Maybe it didn't like that I got on the picture. anyway. So you got that string of pearls with the G actin, and that string of pearl twisted on itself is the fibrous actin, F actin. Okay, fibrous actin. Okay, and on it, you know, when you twist a necklace of beads on it, you'll see grooves spiraling around it like that, like a helix. Around that groove will be another fibrous protein, a string-like protein that wraps around that groove, and it covers the binding sites on actin, and that is tropomyosin. Okay? And for tropomyosin to stay in place, okay, there's another 3 globular subunit protein called troponin, which anchors tropomyosin onto actin, onto the beads. Okay? So when calcium comes in, when the muscle is contracting, it will attach to troponin, because calcium is charged, and proteins are also charged molecules. It's going to make a different attraction so that it changes shape. When troponin changes shape, because it is bound to tropomyosin, it's going to move tropomyosin out of the way and expose what? The binding sites. See these yellow bits? Those are the binding sites. Oh my God. Okay, good. So the tropomyosin covers the binding sites, and I'll show you this picture a couple of times, several times actually. And then the tropomyosin, the red one, okay, does what? Binds the tropomyosin onto actin. So, and the way they are arranged is that at the Z line here, see right here, the Z line, okay, that's where actin is attached on one side, and from here to here, from one Z line to one Z line, is the contractile unit called the sarcomere. So that's the smallest functional unit of a muscle that can contract, okay, because of the way the actin and the myosin are arranged, right? So the myosin, which forms the A band from here to there, that's the A band, right? Okay, and it is anchored here at the M line by a protein called desmin. Okay, and the Z line has a protein called connectin that anchors the connectins, and here is desmin. And I'll show you this to you again later on. Okay. So when the muscle is contracting, what's happening? The Z line is moving in closer, and this one is moving in closer to the myosin, so that this I band is going to start to disappear, and this H zone here in the middle is going to start to disappear, and the sarcomere is also going to get shorter, right? In fact, the muscle is fully contracted when it reaches, the Z line reaches where the myosin is. See what I'm saying? And I'll show you this picture several times. Okay, and then the actin myosin is, the myosin is connected to the Z line by this protein called chitin. It's a kind of springy protein. But myosin doesn't move. All it does is do this with the ratcheting action of this. And I'll show you a cartoon where you can see that. And then it pulls the actin inwards. Okay? So on the actin myosin, on the actin molecule, I want you to remember the tropomyosin and what the function of it is, which is to, what is the function of the tropomyosin? This blue protein. Cover the binding sites, right? And the function of the red 3 globular units here is to, it's binding site for calcium, but what does it do? It anchors tropomyosin onto actin, right? And when it attaches to calcium, that's when it changes shape and moves tropomyosin out of the way to expose binding sites. Because the myosin cross bridges will bind as long as the binding sites are exposed. So when you don't want the muscle to contract, you hide the binding sites. All right? Okay. So, sarco is a Greek word meaning flesh. Okay? Sarco is a Greek word meaning flesh. And so anything to do with the muscle fibers, which is a muscle cell, right? Like the cytoplasm of muscle cell is going to be called the sarcoplasm. Okay? The endoplasmic reticulum of a muscle cell is going to be called sarcoplasmic reticulum. Okay. The only thing about the that's kind of different is the plasma membrane of a muscle cell. It's called the sarcolemma. Okay. Again, the word sarco is what that means flesh, which stands for muscle, and that's what it is. And then of course sarcoplasm. So another prefix is myo, like the myosin, or myocardium is the muscle of the heart. All right. And mice, like the mycium, like the endomycium, epimycium, all those. All right. And then what else? Sarco, we just did that one. Okay. Meaning flesh. So myo is the muscle, means muscle, and this means flesh. Okay. And so the actin and myosin are called myofilaments. And we said actin and myosin are nothing but organelles of the muscle cell or muscle fiber. So that's all it is. And they're called myofibrils. And they are pegged in such a way that the actins slide over the myosin. Okay. One important thing to note is that the actin itself, the myosin itself, when the muscle is contracting, do not change length. Okay? They remain the same length, they just slide into each other, that's all. They don't change length, they remain the exact same length, because you can't really create or destroy the net, it still remains the same length. They're not like elastic that, like the transitional tissues and so forth. No, they just slide into each other, but they remain the same length. Okay, so that's the important part. Okay, most of the volume, of course, of the muscle cell or muscle fiber is made up by those actin and myosin. That's why you have the nucleus on the outside. There's no space for it. Besides, it's going to hinder that sliding mechanism between the actin. Right? It won't work. All right. The muscle cell is packed full of these contractile proteins, which don't change length. They just slide past each other. Again, here's our favorite picture. I show it to you now, blown up. So from one Z line, it's called Z line for obvious reasons. Right? It looks like a zigzag. So Z line to Z line is the sarcomere, and then the Z line is attached to by the connectins to the actin. See this actin, these beads with the globular and the fibrous and whatnot, with the binding sites. And then, of course, the myosin is anchored in place. So the myosin doesn't even move. That's why from here to here, forming the A band, that doesn't change length. It stays the same the whole time. So when I say which of these bands change length, then I'll say I band, A band, H zone. A band doesn't change. It stays the same. It's only the I band that's going to disappear when the Z line is moving towards the myosin. Okay? And it was in your module again in terms of describing this I band. Okay? It started the Z line and ends where the myosin ends are located. See, the myosin ends are located here and here. So that's where the I band is located. So this is the I band. All right? And then when the actin ends, okay, leaving only the myosin is what is called the H zone. Okay? And you can see when the Z lines are moving closer together, this is disappearing. In fact, when the muscle is fully contracted, the H zone is completely disappeared. All right? And the I band is also almost completely disappeared too. Because the Z line will be right there by the myosin. So there will be no I band. The A band is going to still remain the same length from here to there. Okay? Again, let's go over our proteins on actin. So we get our string of pearls twisted on each other with the beads forming the globular actin. Okay? So those are the globular subunits. Right? And then around it will be the blue protein called the tropomyosin. And the tropomyosin is just a long, lengthy protein that covers the binding sites. Okay, and for it to stay in place, you need a 3 unit. Okay, those 3 units, 1, 2, 3, that will bind and anchor tropomyosin onto actin. Okay, so that's why when the calcium comes in, it will attach to troponin, and then when troponin changes its shape, it's going to pull tropomyosin out of the way with it. Okay, and then the binding sites are exposed, and as long as the binding sites are exposed, those cross bridges will form. Okay, that is when the myosin heads attach to the binding site on the actin. Okay, and then when that happens, that takes energy, and then they contract because they are extensible. It takes energy to break the bonds again between the actin and the myosin, and then because they are elastic, they spring back. Then another ATP comes in, and then causes that. So that was in your module when they were talking about what is ATP used for the muscles contracting. Okay. So it's used to attach the protein to break the bond between myosin and actin, and to contract the myosin heads, so that the ratchet, in a ratchet-like motion, will be pulling the actin inwards, and you will see the cartoon. Okay, and that's that. So, and this one is just that the myosin heads and the hinge, which will allow it to move, right? And you can see how we start with the epimysium and so forth on the outside, and then the fascicle, which is a bundle of muscle fibers, and then here you pull a muscle fiber. Of course, you have the connective tissue on the outside, which is the endomysium, and then inside of it you have these bundles of myofibrils, actin, and in fact, around one thick filament myosin, you have 6 actin surrounding it, and I'll show you a picture in a little bit. All right. See our favorite picture keeps popping up. This time it's all bound up into one. So we get the actin myofilaments, right? Alpha helix. Well, it's a helical protein of globular subunits wound around with the tropomyosin right there, covering the binding sites attached by troponin. Right? And then we just said the F actin is that string of pearls or beads twisted around each other, and it forms a nice fiber, a helical fiber. And then the globular subunit, globular subunit, globular subunit, globular subunit. That's it. So that's the G actin. And the troponin is the red one, the 3 subunits. Right? The blue one is the tropomyosin, which is a fibrous protein. So be able to describe the structure of the protein and the function of that protein. One, the actin. Actin is a thinner filament, but it's the busiest. It's a lot busier than myosin, the thick filaments. Because all the action is going on the actin, the thin filaments. Because the actin has the active binding sites. All myosin does is that when the binding sites are exposed, it binds. That's it. And then contracts. Binds and contracts. Until the muscle is fully contracted, or you remove the calcium. When you remove the calcium, troponin goes back to its original shape, the binding sites are no longer exposed, and muscle contraction stops. So that's why at the end of exercise, when your muscle is getting tired and it's not clearing all the calcium out, you get what's called the Charlie horse thingy, when your muscle remains contracted and it's painful. That's what will be happening, because your body is now tired, because it takes energy to clean up all that calcium and to dump it back into the sarcoplasmic reticulum, right? Okay? So you get a Charlie horse towards the end of your exercise when you're now tired and you're leaving calcium in there, and then the calcium is causing your muscle to remain in a state of contraction. That's what becomes painful. Because if the muscle remains contracted like that, it's going to denature, which means it destroys the structure of the proteins. That's why when you die, you get rigor mortis. Not that I want to do that, guys. Yeah. You get rigor mortis, and then you're stiff, and after a while, when the proteins have denatured, you're just like, you know what I'm saying? That's what happens. It's because of these proteins. All right. So, know the tropomyosin, know the actin, know the troponin. All right. tropomyosin actin, the globular actin with the subunits with the active site, binding site, or the myosin binding site. Okay, so because sometimes I ask you to describe these proteins on actin, and tell me what their function is and their structure. So, and for you to understand muscle contraction, which follows this lecture, you really need to know these proteins and what they do. Because then it's all going to be based on that. So the thick filaments, like I said, called myosin, pretty much just have the myosin heads and a hinge here to allow it to move like this, and they just pretty much contract when they are attached to the actin binding sites, and and that's it. So they just have the head, which forms a cross bridge. Once the actin and myosin are bound, that's a cross bridge formation. And we'll talk about cross bridge cycling when we do contraction, muscle contraction. Okay, and then of course the head, and then it's got that hinge, and then it's pretty much a bundle of these myosin proteins, and they're thick, and that's it. And then it's like a rod shaped. So, and what does it have? This enzyme. What does this enzyme do? Just by the name of it, breaks down ATP, yes, breaks down ATP. So the head of the myosin is that ability to break down the ATP, which is used during the contraction and also during the breaking down of the cross bridges. So cross bridges is formed when the myosin head binds to the actin binding site, which is the active site of G-actin, the globular unit of actin. So this is the cartoon I was talking about. I hope this is all.
Myofilaments are composed of strings of the proteins actin and myosin. An actin filament has two strands of actin molecules wrapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
Okay, and it contracts like that. So you see the energy, how it's used up in terms of doing the contraction and breaking the bond between, and then finally to move the calcium back into the cytoplasm. That takes the energy. So that's why when you're tired, you leave all the calcium outside, the binding sites will remain exposed, and the muscle will remain in a state of contraction. It's going here. Not good. All right. Okay, so moving right along. So there you go. So remember, like I said, from one Z disk to the other Z disk is the contractile unit or the functional unit of a muscle cell, is the smallest unit or the smallest section that can contract. So it's called a sarcomere, and it starts from one Z disk to the other Z disk. All right, and then you have your from here to there again is the I band. All right, and then from here to here, that's the H zone, right? Okay, and then what is it from here to there? What's that? The A band. Okay, and the A band is the only one that doesn't change length, right? You can see how that, because the actin is anchored here by the desmin proteins. Right? So the A band doesn't change length. The I band from here to there changes length when the Z line moves and approaches the myosin heads. I mean the myosin, the ends of the myosin molecules. Okay? And this is titin, which is the elastic protein that kind of sort of attaches myosin to the Z line. Okay? So you pretty much know how the A band comes about, the I band, the A band, and the sarcomere, the unit, all right, and what changes length when the muscle is contracting, right, and what doesn't, because I'm going to need you to know that, all right. And then the actin and the myosin are the myofilaments, in the myofibrils. So they appear striated with these bands. Okay, so that's it. And again, this is a picture showing you sarcomere from Z line to Z line, right? And then, the I band on either side of the Z line, okay? Because this continues on, right? And so forth and so forth. And then you can see the M line right in the middle. Okay. All right, so thick filaments, which is the myosin, that's the length of the A band. And the A band does not change length, because it's the length of the myosin molecule. And we said myosin and even actin do not change in length when the muscle is contracting. They simply slide past each other. In fact, the actin is the one that's sliding past the myosin, because the myosin, remember, is anchored in the middle by those desmin proteins at the M line. So the myosin is not even moving, except when the head does this. That's all. Otherwise, the myosin is staying put right in place. Right? So the thin filaments are the ones that are moving in, and then of course the I band, which starts at the Z line and ends where the myosin molecules start, and so as the Z line moves closer and closer to the myosin, the I band disappears. All right. Okay, and then the Z line is got disc-shaped proteins called the connectins that connect the. Makes sense, right? It's easier to remember that. Connectins right at the Z line connecting with the actin. All right? So you've got actin filaments on one side of the Z line, actin filaments on the other side. And then you've got sarcomere. And then sarcomere is the functional unit of a muscle cell. It's the smallest unit that can contract. All right? And then, of course, the H zone is where you just have only myosin, right? Because the overlap between the myosin and actin falls short. It doesn't go all the way, right? So that part where you just have the myosin molecule is the H zone. And the H zone disappears as the actin moves inwards towards the center. In fact, when the muscle is fully contracted, the actin molecules overlap just slightly, and the H zone is completely disappeared. Okay, it's not even there. And I'll show you the progression of the contraction in a picture and visualize that. So the H zone definitely changes length, and we talked about it. A protein called desmin attaches the myosin molecule right there at the M line, and then the connectins at the Z line. Okay? So again, know these proteins and know their function. Know all the proteins on actin and their function and structure. Okay? Alright? And the bands, know which bands, what they're made up of, whether it's just actin alone or just myosin alone, and whether they change length when the muscle contracts. Alright? Arranged nicely, of course, and then because of that overlap and so forth, creates that dark light bands, striations. Right? And then again, our next picture of the striations is skeletal muscle fiber. You can see how the nuclei are located on the outside. Remember, like I said at the beginning, you have 6 actin, 1, 2, 3, 4, 5, 6, surrounding a myosin molecule, right? And then you see these cross bridges, these are heads, myosin heads. Those are the ones that are attached to the actin fibers. So you can see each of the sections, and like this is the section that you can see the overlapping. So in fact, this will be the darkest section. The darkest bands will be where the myosin and actin overlap. The lightest bands will be where the thin filaments alone are located, which is the I band, right? This one will be lighter, but because it's the H zone, but because it's made of the thick filaments, it's at least darker than the I band. So that's why you have dark bands, light bands, medium bands, and that is what creates the striations. All right? Did you see that? All right, very good. All right. So when those actin and myosin filaments slide past each other, well, with the actin slides past the myosin, it's the sliding filament model, right? Actin and myosin filaments will slide past each other, but they do not change length. Like I said in the beginning, they remain the same length. They just simply slide into each other, and the degree of overlap increases. That's it. And when the muscle is fully contracted, they are fully overlapped, and the myosin and the actin are touching in the middle. In fact, slightly overlapped. All right. So upon stimulation, you have the impulse comes in, and the impulse opens the calcium gates so that the sarcoplasmic reticulum. So that process doesn't take energy; it just takes the stimulation, because it was one of the choices in your list of the module. It was like, does this process take energy? It doesn't take energy. All it does is take the impulse to come in, and then open the calcium gates, and the calcium will come out. It's only when you have to put the calcium back into the sarcoplasmic reticulum that you need energy. Okay? You need the ATP for that. So, and then the calcium comes out, will attach to troponin, and then it will change its shape, and in doing so, move tropomyosin out of the way, and then the binding sites are exposed. And as long as the binding sites are exposed, those myosin heads will bind and contract using ATP, bind and contract, and bind and contract, and until the muscle is fully contracted, or until you clear the calcium. Okay? So that's what happens in a ratchet-like motion. And then the thin filaments will be the ones that are moving inwards. The thick filaments stay put to the myosin because they are even attached right there, the M line by the Desmond project. Alright? So, relaxed, the overlap is slight. It's not full. Completely contracted, the overlap is at maximum, it's greater. The overlap between actin and myosin. In fact, the muscle will be, the sarcomere will be the length of the actin molecule, right? Because the Z line will approach the myosin molecules. So, and it will stop right there when it gets to the ends of the myosin, and then the muscle is fully contracted. All right. And this occurs throughout all the sarcomeres, and they all shorten, and the whole muscle shortens. So, I band, H zone, sarcomere shorten during muscle contraction. The A band stays the same length. Okay. Okay. So those are the only things you need to know about. And like I said, this is showing you the degree of overlap is getting higher and higher and higher. It gets greater and greater until here, when you see a slight overlap over here, okay, between the actin and the myosin, and the muscle is fully contracted, and you can see how the Z line is approaching the myosin, and you have a just thin sliver of the I band remaining. It's almost also completely disappeared. But the H zone does definitely completely disappear. And so the following pictures pretty much show the blown up part of that 4-picture diagram. All right. So that's the sliding filament model. It's pretty much the actin sliding up on the myosin. All right. So this is the relaxed muscle. There's degree of overlap. Okay. That is just right for the muscle to contract. That's why sometimes when the muscle is overstretched, the degree of overlap is going to be almost zero, and then the muscle won't contract with enough force. That's what they call the length tension, effect. It forms kind of a graph like that. So this is the degree of tension, and this is the length. Okay, so right here is the peak amount of overlap in this relaxed muscle where you get the maximum tension. If it's too much it's squished, there's not enough distance to move, so the force is less, and then it goes down. If it's too much stretched, then the muscle will contract with less force, because you need some degree of overlap so you can have these myosin heads are touching. Right? Makes sense? Okay, so you start off relaxed and then you slowly move. So like I said, these are the pictures shown on that first slide with different degrees of overlap as the actin molecules move inwards, thereby making the H zone disappear and making the I band also smaller and smaller until it almost disappears. Right? So the sarcomere also gets shorter as the Z line moves closer to the other Z line, and the other Z line is also moving closer to the others. Alright? So this is pretty much it. Sliding filament model, and here we are almost fully contracted, and you can see how the H zone is really very small now. Okay, and the myosin is still same length. Myosin is anchored in place, right? Okay, so the A bands remain the same length, don't change, right? And then now you can see the muscle is fully contracted, and you have even a slight degree of overlap right there. So the H zone is completely gone. Okay, all you have is a sliver of I band remaining, and the muscle approximates the length of which molecule, which protein. At this stage, when it's fully contracted, the muscle sarcomere is the length of. The actin filaments are the blue ones, but it's approximately length of myosin. You see, myosin is from here to here, because actin is only from here to there. Okay, so it will take two to span the length of the sarcomere in this case. See what I'm saying? So, but you see, from here to there, it's all myosin. So when the muscle is fully contracted, it approaches the length of myosin, the thick filaments. Okay. Because I may have like a question like that will kind of trick you.
Can you also say the A band?
What's that?
Can you also say the A band?
That's the A band, right?
Can you say the A band?
Oh yeah, you sure can. Because the A band is pretty much the length of myosin. That's why it doesn't change length. And in the first place, we say myosin and actin do not change length when the muscle contracts. Okay. All right. So this cartoon, we could watch it though. Since we have how many seconds? We have a few seconds.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament. As a result, the sarcomere is shortened. In the fully contracted muscle, the ends of the actin myofilaments overlap. The H zones disappear, and the I band becomes very narrow.
Yep. So that was short and sweet. I should have watched it in the morning, but I had gone over a little bit. So again, as usual, this is just a table that will compare and sort of talk about what these interactions between the organ systems is all about. Okay. So thank you guys. I'll go ahead and finish the grading, and then I'll give it to you in the lab real quick. Today we have our last review session before the practical. So I want to do that real quick and give them back to you, okay? So we start at 5 because I want to finish grading the exam. Okay?